Hello everyone, I'm Aurchie Rahman. I'm a graduate student at the Bioanalytical Lab of Dr. Saiful M. Chaudhary at the Department of Chemistry and Biochemistry at UTA. My poster is entitled Proteomics Study Approach to Discover NLRP3 Inflammasome Protein Interactors. So the research is discussing about the pattern recognition receptor NLRP3. NLRP3s are cytosolic receptor which sense dangers to the cell. So whenever there is some cellular damage happening to the cells due to maybe some metabolic disorders or some nervous system disorders, or if there is some inflammation caused by some microbes to the cells, so there is overexpression of this cytosolic receptor NLRP3. So NLRP3 proteins are then getting together with adaptor protein ASC and with the procaspase 1 protein, which acts as an effector molecule. So they get together and they form this oligomeric complex called NLRP3 inflammasome. This inflammasome then creates some active form of inflammatory cytokines IL-1 beta and IL-18. The study of these proteins are important because they can help us design better drugs to treat several metabolic disorders, several nervous system disorders, and some inflammatory disorders as well. So to study the proteins that are interacting with NLRP3 receptor, we studied our work with the culture of monocytic cell line THP1. We created samples of LPS treatment, of nigiricin treatment, of LPS and MSU treatment, and of LPS and nigiricin treatment. Then we created cell lysate samples. On the samples we did in solution digestion technique. We purified our protein samples with methanol chloroform precipitation technique. We quantified our proteins in the samples by performing BCA assay. And then we put our digester samples to the LC-MS-MS analysis. After the analysis were done, we created the database search and we found the proteins that were expressed in all these different treatment sets. We tried to identify their function by performing some bioinformatic analysis on the identified proteins. So we noticed when we were doing LPS treatment and nigiricin treatment on the cells, there were some pyroptotic cells in the sample. So cells were dying. So definitely there was activation of the NLRP3 inflammasome complex. So when we did the quantification of the identified proteins, we saw there were around 3,000 proteins expressed in the nigiricin treated cells, and there were around 2,500 protein IDs in the LPS and nigiricin treated cells. So when we created Venn diagrams, it helps us to identify which proteins were commonly expressed in some treatments and which proteins were exclusively expressed in some treatments. So when we did the Panther classification on the identified proteins, it gave us the molecular function, the biological function, and the cellular location of these proteins in the system. So we saw that the identified proteins were mainly participating in catalytic activity and in binding activity in the cells. So the study gave us exclusive proteins which were important in the NLRP3 inflammasome activated cells. So we hope to validate our data in future by performing some Western blotting and by performing some fluorescence studies. So in our study, we want to acknowledge the funding from National Institute of Health and National Science Foundation. So this research hopefully will give us some insight on the metabolic disorders and the nervous system disorders that is affected by activation of NLRP3 inflammasome complex. Thank you for your patient hearing.